previous part, we have reviewed three fundamental methods of the experimental structure determination by biomolecules. These methods are X-ray crystallography, NMR, and cryo-electron microscopy. We also practiced 3D structure retrieval from protein data bank and graphical rendering of those structures using PyMol. Note that currently PDB has more than 160,000 structures deposited. Some researchers believe that at least one representative structure of the existing protein fold families, estimated to be in the range of 3,000, has already been solved. Can we use this massive amount of structural data and algorithmically predict all the needed structures? It seems we are rapidly approaching this situation. The explosive development in the area of artificial intelligence and interests of industrial giants like Google, Microsoft, and IBM might make this vision very real in the immediate future. Let us watch a short video discussing the technological breakthrough in the area of protein structure prediction.
Yo, this protein needs to fold faster. Add some AI to it. Hello world, it's Siraj, and last month an AI lab called DeepMind entered their algorithm dubbed AlphaFold into a competition to predict the shapes of proteins and won. This is a big deal. Unlike the rest of the teams, DeepMind had zero experience in molecular biology, yet they still beat top pharmaceutical companies with 100K plus employees like Pfizer at this challenge. And not only that, they won by an unusually large margin over second place. DeepMind demonstrated that an engineering solution, not necessarily a breakthrough insight, could accelerate scientific discovery. Their convergence will dominate the 21st. AlphaFold is the perfect example of a convergence between two, biology and information, the gene and the byte. A gene is a region of DNA that encodes some instructions. DNA is the blueprint for all living things. A copy of it is found in every cell of an organism, from a strand of your hair to your moneymaker. Think of it like software, and the cell with its molecular machinery is the hardware running the software. DNA needs a living cell to carry out its instructions. In the proper setting, these instructions are powerful, producing a whole human being from just a single cell, as it did with each of us during our 8 months of gestation in the womb. DNA's instructions are used to make proteins, the complex molecules that do most of the work in our bodies. The instructions of DNA are transcribed to an intermediary molecule called mRNA. The information contained in RNA molecules are translated into the language of amino acids, which are the building blocks of proteins. This language tells the cell's protein-making machinery the precise order in which to link the amino acids to produce a specific protein. This is a big task because there are 20 types of amino acids which can be placed in a whole lot of different orders to form a wide variety of proteins. These proteins fight viruses, transmit signals to coordinate biological processes, provide structural support for cells, transport molecules, act as storage devices, make you swole. They have a huge range of functions in the body. Proteins are complex molecules. They aren't stable in their unfolded or fully extended shape. They spontaneously fold inside and outside of the body based on interactions between amino acids and different environments. The ability to predict a protein's shape allows us to better diagnose and treat diseases that are believed to be caused by misfolding proteins like Alzheimer's, Parkinson's, Huntington's, and cystic fibrosis. Knowing how proteins operate opens up new potential within drug discovery. It will enable scientists to create much more effective cures for diseases more efficiently, which would ultimately improve the quality of life for millions of patients around the world. In fact, any material with any set of desired properties could be constructed using proteins. We could build a material as hard as a turtle's shell or as soft as a jellyfish. We can build liquids that dissolve plastics or the most flexible fibers in the known universe. Think of proteins like generalized materials. If we understood the inverse function of material properties, meaning their protein structures, it would change the world far beyond the monetary value of the invention. We could improve crop efficiency by creating insecticidal proteins or frost protective coatings. We could regenerate tissue in disabled veterans through self-assembling proteins. A whole new realm of cosmetic and enhancement supplements for improved health could be introduced. Solving protein folding doesn't just mean curing diseases. It means helping solve world hunger, enhancing our own physical capabilities, and creating new biomaterials. A protein starts off as a straight string of amino acids chained together in a unique ordering. Then, an initial folding process occurs that includes a few twists and zigzag folds, giving it more of a 3D shape. This folding progresses as component amino acids come together through attraction across disparate lengths of the protein, and sometimes the resulting protein will attach to other proteins after finishing folding in order to function properly. This whole process is driven by energy minimization. We can imagine the process of protein folding as a hiking expedition across a field of rolling hills to find the deepest valley. The tops of hills describe protein folds that are very unlikely to occur, and the valleys describe states that the protein is drawn towards, the native state. For a given protein of X amino acids, there are about 3 to the X possible folds. This quickly becomes intractable for proteins of meaningful size, which contain hundreds of amino acids. It's a high-dimensional landscape. Trying to predict the shape of it remains difficult for most methods. Thus far, scientists have been able to predict some protein shapes using experimental techniques like cryo-electron microscopy, nuclear magnetic resonance, and X-ray crystallography. Each of these methods is a time-intensive process. Lots of trial and error are involved, and it often takes years and tens of thousands of dollars per structure. But the rapidly dropping cost of genetic sequencing has increased the amount of available genetic data to train on, and this is a major reason why biologists are turning to AI as an alternative to traditional prediction processes for difficult proteins. CASP, the community-wide experiment on the critical assessment of techniques for protein structure prediction, was established in 1994 and became the gold standard for assessing different protein folding prediction techniques. It's helped catalyze research and measure progress on the newest methods for improving the accuracy of predictions. DeepMind entered into the competition and won against a total of 98 competitors, vastly outcompeting the team winning the second place. DeepMind be like, get wrecked. While the team in second place could predict only 3 of 43 proteins, AlphaFold correctly predicted the structure of 25 of those. The protein folding problem has been worked on now for over 5 decades. The challenge is that DNA only contains information about the sequence of a protein's amino acid chains, its building block. Predicting how these chains will fold into the intricate 3D structures of a protein is the hard part. Over the years, as scientists have been able to discover various protein structures using different techniques, a database of proteins was compiled called the Protein Data Bank. It's a dataset that contains over 146K proteins, and DeepMind used 29K of them to train its neural network. The input would be the amino acid sequence, and the output, or label, is the collection of coordinates that represent the resulting protein's 3D shape. Yay, supervised learning. AlphaFold was able to learn the relationship between the amino acid chains and their resulting protein structures. Then, given a new amino acid chain, it could predict an associated protein structure. AlphaFold consisted of 2 deep residual convolutional neural networks. We've seen a lot of advances in tasks like image recognition and speech recognition by simply stacking more layers onto neural networks. The more layers a network has, the deeper it's considered. But simply stacking more layers onto a network only goes so far, and after a certain threshold, accuracy starts to drop. Microsoft Research thought of a brilliant solution to this problem. Sorry about that. Just connect the output of previous layers to the output of new layers. Assuming you have a 3-layer network, for example, you wouldn't only pass the output of layer 1 to layer 2 and so on, but you would also add up the outputs of layer 1 to the outputs of layer 2. This residual architecture helped them win the ImageNet competition in 2015, and DeepMind repurposed it for protein structure prediction. Their 2 ResNets predicted the distances between pairs of amino acids and the angles between the chemical bonds that connect those amino acids. They used the popular gradient descent optimization technique, using a combination of these predicted features to optimize full simple protein chain structures. They input the results of their predictions into a 3D modeling software suite called Rosetta to help visualize the resulting proteins. Rosetta is able to construct a protein structure in both Cartesian and internal coordinate representations using these features. In another method, they used a generative network called a direct recurrent attention writer, or DRAW, but it's not clear how exactly it was used, so we're not going there right now. I also have a great video on how gradient descent works, by the way. Link will be in the video description. So that's AlphaFold. It used this dataset to generate new structures from a given set of amino acids and was scored on its accuracy using 2 deep residual networks. Demis, CEO of DeepMind, stated that they didn't solve the protein folding problem entirely, but it was a first step. It's likely that the next step for them is to incorporate reinforcement learning into the pipeline to help increase AlphaFold's accuracy. We have no idea how much computing power was used here, but I think it's fair to say a lot.
Amazing, isn't it? The newcomer in the academically established field of the protein structure prediction, DeepMind AlphaFold, has won the competition by a large margin. The lesson for us is we have to stay tuned and be ready for a new bioinformation revolution. What kind of problems already existing predictors can solve? Recall the J-coupled protein receptor from the video Inner Life of a Cell. What if a protein I am interested in is functionally similar to it? Say it might be involved in signal transduction across plasma membrane. Can I just predict its structure, predict its potential ligands, and from there its function, and come up with the limited set of mutations which would confirm my model? There's going to be publishable data. In the following, I will show you the practical workflow of such a prediction using one of the publicly available web services. It is not AlphaFold, since it's of now, it's not freely available to public. I will use the web service called iTasser, which is still the second runner-up in the CASP competition. Here is the example of the protein which I wanted to predict in the course of my genetic studies. So here is the novel gene was discovered, which controls the behavior of the mouse. So in a nutshell, there are 2 types of the mice, like this, which are actually subspecies, and then this deer mouse, and the fur differs in color. So here is the light, and then here is the darker. And then they also differ in their behaviors. So this lighter colored mouse, they built additional escape tunnel to their burrowing. Like here, this deer mouse, the darker in color, so has no escape tunnel. So then it would be quite interesting to see what's the genetic difference between them, which might control this behavior as well as the color of the fur. So after the crisscrossing, so these mice, they managed to localize quite short, quite small, compact genetic locus, and after additional research they localized this gene, MC1R gene, and they state this may be linked to mouse color and behavior. So that sparked my interest. I started to look at the mouse genetic information database to check whether this gene is really related to the behavior. And so here it is. This gene was linked to pigmentation, nice, and also linked to behavior. So I was pretty sure that I'm on the right track because this gene is involved in these 2 critical phenotypes. So I decided to predict this, and my first task was to retrieve the amino acid sequence of the product of this gene. So therefore, I went to the NCBI dataset, so web service, and then I selected gene database, so you can pull it right here, and then I gave a query, the name of the gene and the organism. So this was the return. of my query, and this is indeed MC1 receptor, and coming from the house mouse. So next, I clicked on this link, and I wanted to see the genomic localization of this gene. The point here is like that. So some genes might have a different copy numbers. So if there are different copies, so several copies of this gene, so I have to then be more careful which one to pick up. And I looked at the genome localization. Here it is. So showed with one red arrow. So it means there's a one gene copy number. So I can proceed to analyze the isoforms of this gene. So what are isoforms? DNA transcribed to mRNA, then mRNA is spliced. It can be alternatively spliced, and therefore several transcripts might appear right here, and then they might result in the translation into different amino acid sequences. So therefore I have to choose the right isoform. So typically they appear in they transcribed in different tissues. So you need to know more about biology. Here there's only one transcript, so I proceeded. So I scroll down and till the mRNA and protein sequence here. So here is the mRNA sequence, and in blue the protein amino acid sequence product of this gene is shown. So I clicked on it, and then this brings me to the page describing this protein, and there's on the right side a menu called conserved domains. So you have to click absolutely on that one, and this gives you the synopsis of what is available in terms of the structural information regarding to this amino acid sequence. So here what do we have? Here we have a total sequence of the protein. Then we have this identification of superfamilies, and then here we have a domains. And this sequence I have chosen for the prediction. So I have to remove the flanking sequences because very likely they are not structured. So, and therefore if you add non-structured sequences to the prediction, so you total score, confidentiality score will drop down, and then it might drop down below acceptance. So that is the point. So I have to predict only conserved domains or recognizable domains. So here is the full sequence, and I decided to predict the range from the 50 to 300, which corresponds to the 7 transmembrane domain of this protein. So this sequence part is highlighted in red. So then I submitted this sequence to the iTasser. So in the future, might be just AlphaFold would be a reasonable option, but currently iTasser is the second runner-up. So I just pasted it right here, so in this window, and pressed go button, and depending on the server load, but usually it's overnight, so I got result. The result came as a collection of 4 models right here with associated C-score. C-score is a confidence score. Basically, it varies from -5 to +2, and anything above 0 is considered as acceptable. So here, my model 1 gave me kind of the highest score, so I picked up this model and proceeded with the analysis. How I can assess the resulting structures. So I can superimpose in PyMol, right here, PyMol. I can align my model, which is this one, so with the templates they used to predict, and then I can check the assessment level, so how closely my model fits to the template. So here we see that transmembrane part of the template is blue, and this is my model. So for transmembrane part, here is the membrane approximately, so fits pretty well. So it gives me kind of like a good feeling that I'm on the right track here. So then I move on, and then I checked against the second template, which is actually beta adrenergic receptor right here. Yep. So again shown in blue, and then transmembrane part like this, fit or align pretty well actually. So in here, this is the functional part. All right, so now for the safety, I predicted this model also using another server, which is actually Raptor X. So for some people, it's choice number one. And the resulting models are pretty much the same. So the next, to predict the function of this protein. So I wanted to compare, to align this protein structurally with other proteins available over there. So for that, we have to use DALI server, so available through this link. You submit, here it will be clear. So you submit your sequence from here, from this button. You submit your structure, your PDB structure. In this case, it's going to be model. Yeah. So, and then it returns structural homologs found. What I found here. So here, this is the output of the DALI. So this is my query model. And these are the structural homologs. So the proteins which structurally resemble my query. And here I can overlay them. So this is again the output, direct output from DALI. And what I see here, so they are very similar proteins structurally, yeah? Especially in the area of the membrane spanning helices. And here, so are the ligands which are found in the closely matching proteins. So I see that they are the area where the ligands bind is very conserved. The other structural components are quite conserved along them. So they gave me the confidence that my protein might be one of these kinds. So now I can look what these proteins are doing functionally. So these are these proteins. And immediately what became very clear from here is that they all respond to hormones of adrenaline, or adrenaline derivatives, so all of them. So from that I inferred that perhaps my target, so melanocorticin receptor, might be also responsible to adrenaline. This gave me kind of like an outlook on the function. So perhaps the mice with the lighter 4 might be more anxious, so having higher response to adrenaline, and therefore like burrowing, and the other one more relaxed mouse, and just simply dropping the burrowing after the main nest is made. So that is the analysis. So basically this gives a workflow of the analysis, and in the application exercise you might just follow this workflow in analysis of your own protein. All right, let us quickly recap. So the most reliable predictions can be done for the compact domains of the proteins. So you have to identify in the sequence the compact domain, make sure that it's conserved, so there's a homology to it, and then you can submit the sequence corresponding to this domain to one of the prediction servers. So that is straightforward. So the tricky part is to analyze the result which come out of this server. However, what if your protein is not monomeric? So the compact domain might associate into the homo-oligomers. Homo means that they are the same, but the same domain structures just come together and form homo-oligomers. So the number can be different, it can be dimer, trimer, and then so on. Like that situation, then can we model the structure in this case? That situation has exactly happened in my own research of the ion channel from SARS-CoV-2. So here is the PDB entry of our structure right here. So, and it's a SARS coronavirus protein, but it is pentameric. ion channel. So it consists of 5 homo-oligomers. So can we can look at the structure. So here it is, and you can see we can rotate it around, and here in the middle there's a hole, so the ions are passing through this hole, and it's pentameric. You can see 1 monomer, 2, 3, 4, 5 pentameric channel. Just to give you some background on that protein in here. So here we have a membrane like this, so it's spanning the whole membrane. Okay, can we model this structure in particular? Yes, we can. So we have to use a special server which is called Galaxy Homomer. So it's right here, Galaxy Homomer. There's a publication, so the link to the publication and to the server itself is in the description below. So the procedure is straightforward. Actually, you have to just submit one sequence and indicate how many identical units should come into the assembly. So this is the interface, and so you can see what you have to provide: job name, email address in here, the sequence, or if you have already monomeric structure, so you can submit this monomeric structure. And then right here, you have to indicate the number of oligomeric states. So 1, 2, 3, and so on. When I submitted my job, I indicated 5 because it's pentameric ion channel. This number of oligomers should be identified from other experiments. Let's say from ultracentrifugation. That's what we did in here. So you submit, and then you got the model, so pentameric model. But what if your monomers are not identical? So you got heterodimer or heterotetramer, and then so on. Yeah. So then in this case, you have to use yet another server, which is right here, which is called Class Pro or Autodoc Vina or some other things, but they are basically the same. So here they say it's a pretty busy server, widely used tool, yeah, for protein-protein docking. So this art of the prediction is called docking. So it's a protein-protein docking. In this case, the monomers might not be the same, so it might be different. And then if I just simply look at their interface, they allow access to their docking server to everybody. And then here you have a receptor, and then you have a ligand, and you can either upload PDB on here and in here, or you can just simply indicate the chain from this uploaded PDB. Then you got a bunch of advanced options in here, but they will definitely be useful. For example, if you know which residues should come together in the structure. But if this knowledge is not available, then leave it blank, and so on. Let me give you an example from our own research. So we have a chaperone, a molecular chaperone, interacting with the beta amyloids in here, and we wanted to dock it. So these two. So we basically loaded everything in this structure and amyloid structure in the Class Pro, and then let it optimize. And it found very specific and interesting interface between these two proteins. So our next step in the research would be then to mutate those residues and see whether the interactions will be broken apart. So that would be then model validation. All right, in a nutshell, the best targets for the structural predictions would be then compact domains, conserved compact domains. Then the homology modeling can be used, and the models will come up reliable. So then if, let's say, the protein consists of the individual subunits, so then you can just basically build them together using either Homomer or Class Pro or Autodoc or any other services which are available out there. In this lecture, we reviewed the details of protein structure prediction based on the homology modeling. In the application exercise, we will apply this knowledge to predict, assess, and present models of proteins relevant to your own research or industrial interests. Together with other skills we have practiced so far, it forms a foundation to address our question of the origin of the novel SARS-CoV-2 virus.